When you think about the food on your plate, you probably don't spend a lot of time contemplating the complex science that goes into it, right?
Probably not, no.
But what if I told you that behind every single bite is this meticulous global system designed purely to keep you safe?
Mhm.
We're talking about the silent guardians who delve deep into the world of pesticide residues.
Precisely. And today, we're taking a deep dive into the latest scientific report from the joint FAO-WHO meeting on pesticide residues, or, JMPR for short. This one's from 2024.
Right.
And our mission, really, is to unpack this pretty dense document, highlight the most important findings, and maybe reveal some surprising facts that impact how we all understand what's safe to eat.
And just to give you some context, this deep dive is based entirely on that comprehensive JMPR report. You should think of it as like a gold mine of information,
It really is.
outlining their detailed evaluations of various pesticide ingredients, everything from their potential toxicity to their exact presence in our food and the environment.
So let's start by understanding the system itself, the JMPR. It's a joint expert scientific committee bringing together the Food and Agriculture Organization, the FAO, and the World Health Organization, WHO.
Okay.
Their core role is quite literally evaluating pesticide residues in food and agricultural commodities.
And to give you a sense of just how formal and globally significant this work is, any disputes that might arise under their licenses are actually referred to UNCITRAL arbitration.
Right, the UN Commission on International Trade Law.
Exactly. It really underscores the weight and, the international reach of their findings. This stuff affects food safety standards worldwide.
What's also crucial to understand, I think, is that many of their summaries and evaluations, they're based on unpublished proprietary data.
Ah.
Yeah, this data is provided by the chemical sponsors themselves and national registration authorities. They actually need specific authorization to use it.
So it's tightly controlled.
Very tightly controlled, yeah. A rigorous process,
Yeah.
ensuring that the raw data is thoroughly reviewed before any conclusions are drawn.
Okay, let's unpack this pesticide vetting process because it's fascinating how incredibly detailed it gets. The first major step involves toxicological assessments, basically, figuring out how much of a substance is too much for us humans. How do they establish those critical safety benchmarks?
Well, they establish safety using key terms like the acceptable daily intake, or ADI,
ADI, got it.
and the acute reference dose, or ARFD. These are essentially the daily or single-dose amounts you could consume without an appreciable health risk.
And these are based on?
They are all based on what's called the No Observed Adverse Effect level, or NOAEL. That's the highest dose at which no adverse effects are seen in the studies they review.
Right, the NOAL. Can you give us a concrete example of how they apply these assessments? Like, how does it work in practice?
Absolutely.
Yeah.
Take a compound like Sononopir. During their chronic studies, they identified a NOAL in rats based on, pretty subtle changes like increased reticulocytes. Those are basically young red blood cells.
Oh.
And some unusual tissue growth in blood vessels, angiomatous hyperplasia. But what's striking is that a separate study in dogs showed an even lower NOAEL, this time due to issues like anemia and certain cellular changes.
So different results in different animals.
Exactly. It highlights how safety levels can vary across species, which really underscores the complexity of setting a truly universal safety benchmark.
It sounds like a thorough, but sometimes challenging process.
Yeah.
Are there ever clear sort of good news stories from these assessments?
Oh, definitely. It's not all about just uncovering potential risks. There's good news, too. Take cyclobutyrophlorum, for instance.
Okay.
The report essentially gave it a clean bill of health, deeming it unlikely to be genotoxic, meaning it's unlikely to damage your DNA, or carcinogenic in mice and rats. So a clear thumbs up on that compound.
That's a relief. But what about like unexpected twists they find? You mentioned impurities before.
Exactly. You get these unique cases like fluozium. Here, white matter vacuolation, which are neurological lesions, was actually linked to an impurity called B1457 in the technical material.
Not the main chemical itself.
Not the active substance itself, no. And this only happened when the impurity exceeded a really small threshold, like 0.20%. It shows how precisely they try to identify the true cause of any adverse effects. Sometimes the culprit isn't what you'd expect at all.
Wow. Now let's look at an example that really highlights these unexpected complexities. Fluoxapiprolin.
Ah, yes, fluoxapiprolin. This one is fascinating. For the parent compound, the main chemical, the ADI and ARFT, are considered unnecessary.
Unnecessary? Why?
Well, presumably the toxicity was very low. But get this. A separate acute reference dose is established for specific metabolites, its breakdown products.
Okay, so the things it turns into.
Exactly. Because these breakdown products, like fluoxypyrazole BDM-pyrazole and fluoxypyrazole carboxylic acid, they have their own distinct toxicological profiles. It's not just the main ingredient they scrutinize. It's everything it transforms into in the body or the environment.
Right, right. And I imagine not every compound makes the cut or, has all the necessary data available.
That's absolutely right. For maleic hydrazide, for example, the meeting actually could not establish an ADI or ARFD.
Why not?
Simply due to insufficient toxicological data. This really highlights that even with all the submitted information, sometimes data gaps still exist, and they prevent a safety benchmark from being set. And the JMPR is transparent about that.
Okay, so from toxicology, we move to metabolism studies. This is all about the chemical journey of pesticides, how they transform and move through plants, animals, the environment. How quickly can these substances change once they're out there?
Well, it varies hugely. A great example of rapid transformation is carfentrazone ethyl. It degrades incredibly quickly in soil. We're talking a half-life, the time it takes for half of it to break down, of less than 1 day. The geometric mean was just 0.013 days.
Wow, that's fast.
Very fast. And while some of its metabolites, the breakdown products, persist a bit longer, the residues actually found in the edible parts of plants were consistently very low, basically less than 0.01 milligrams per kilogram, hardly detectable.
So we've seen how quickly some degrade, but others have very different journeys, right? Take ethoxyquin.
Ethoxyquin is quite unique, yeah. It's highly volatile, meaning it evaporates easily, and it's lipid-soluble, so it dissolves in fats. And its major way of breaking down is through aqueous photolysis. Essentially, it degrades in water when exposed to light. And its breakdown products, like something called DHMEQ, were specifically found in pairs in the studies they looked at. It demonstrates how it changes after application and what residues might actually remain.
And then in stark contrast, you have something like phytoflutophane, which you mentioned is incredibly persistent in soil.
It truly is. We're talking a half-life that can stretch into years. The report mentions DT50 values up to 2,380 days, with a new geometric mean of 521 days.
That's a long time.
A very long time. It raises significant concerns about its long-term presence and potential uptake by new crops planted later in the same soil. The report even tracks a plateau rate it reaches after about 4 years of applying it annually, showing this long-term buildup they're monitoring.
Okay, next up are residue definitions, essentially. What exactly gets measured when they test for a pesticide in food? Is it always just the original chemical?
Not at all, no. Think of it like trying to measure the sugar in a drink. Sometimes you just measure the sucrose itself.
Right.
But other times, if that sugar breaks down into other sugary compounds like glucose and fructose, you might need to measure all of them together to get the full picture of sweetness.
Makes sense.
That's basically what a residue definition does for pesticides. It specifies what needs to be measured. And it can be complex, sometimes involving the parent compound and its metabolites for compliance testing or for risk assessment.
So for something like prochloraz, how is that measured in plants? What's the definition there?
Okay, for dietary risk assessment in plants, the residue definition for prochloraz isn't just the original compound. It's the sum of prochloraz and one of its major metabolites, BTS 44596. Plus, it's also noted as fat-soluble, which is important for how it might accumulate in fatty foods.
What about different rules for different food types, like plants versus animals? Does that happen?
Absolutely. Fenpropidin is a perfect illustration of that. For plant commodities, the residue definition is just fenpropidin, the parent compound. Simple enough.
Okay.
But for animal commodities, meat, milk, eggs, the residue definition shifts. It becomes the sum of fenpropidin and a specific metabolite, CGA 289267, expressed as fenpropidin equivalents.
So different rules.
Yeah, it highlights how detailed the assessment gets, adapting to how the chemical behaves and breaks down differently in plants versus animals.
And fipronil sounds like it's in a league of its own for complexity in this area.
Fipronil truly stands out, yeah. There are various definitions depending on the exact purpose. For calculating the livestock dietary burden for setting maximum residue limits, or MRLs, it's the sum of fipronil and fipronil sulfone.
Okay.
But then for estimating the total residues for dietary intake assessment, looking at the median, STMR, or highest residue HR levels in food, they add even more breakdown products, fipronil sulfide and fipronil disulfonil.
Wow.
It's like this chemical family tree they have to account for, making sure they capture all the potentially relevant forms that contribute to exposure.
And another interesting nuance with spinosad, you mentioned fat solubility earlier.
Right. With spinosad, the residue definition is the sum of two related compounds, spinosin A and spinosin D. But here's the kicker, the interesting part. It's considered fat-soluble for residues found in meat, but not for residues in milk.
Huh. How can that be?
It just demonstrates that fat solubility isn't always a simple yes or no answer. It can be context-dependent, based on how the body processes and distributes the chemical in different biological matrices, like muscle versus milk fat.
Fascinating. And finally, just circling back to maleic hydrazide, was there clarity on its residue definition, even if the ADI wasn't set?
Well, they concluded that the parent compound itself is a suitable marker for enforcement purposes in both plant and animal commodities. So for checking compliance, just measure maleic hydrazide. But the definition for dietary intake assessment, for figuring out the actual risk from eating it, was not concluded. And again, this was due to insufficient data. It's a recurring theme. When the information isn't complete, the JMPR won't make assumptions or finalize certain aspects.
Okay, now let's talk about the human equation. How dietary exposure is actually estimated. This is where the science gets really personal, isn't it? Because it directly impacts what ends up on your plate. How do they measure what people actually eat?
Yeah, this is crucial. They use two primary models for measuring consumption. The Global Estimate of Chronic Dietary Exposure, or GECDE, and the International Estimated Daily Intakes, or IED.
GECDE and I.
Right. And actually, a key finding in this report suggests that using just 2 days of dietary survey data is considered international best practice.
Only 2 days.
Yeah, they found there's no substantial benefit of having a larger number of survey days available for estimating less than lifetime or chronic dietary exposures. So 2 days seems to be enough for that long-term picture.
What's fascinating here, though, is how much these dietary exposure estimates can vary by country. It's not a uniform picture globally, is it?
Not at all. It varies quite a bit. For example, in Italy, the mean estimates they calculated can fluctuate sometimes higher, sometimes lower than the general IED model, but children there often show higher mean estimates than adults.
Okay.
Meanwhile, in Mozambique, their mean estimates are typically higher than the IED across all population groups studied.
And then you have Romania, where the estimates are lower across the board. So what's the big takeaway from all this country-specific data, then?
The big takeaway, and the report states this pretty clearly, is that these general global models like GECDE and IED, they do not capture specific dietary patterns within individual countries.
Right. Makes sense.
The variation of consumption amounts within a distribution, like, people who eat very little of something versus those who eat a lot, or the variation in dietary exposure estimates for pesticides across different countries or population subgroups.
So it really highlights the challenge of creating a truly accurate one-size-fits-all model for global consumption because diets are just so diverse.
Exactly. It's a real challenge.
So how do they handle metabolites, these breakdown products that might not have extensive toxicological data? Do they just guess the risk?
No guessing involved.
Yeah.
They have a clever tool called the Threshold of Toxicological Concern, or TTC.
TC. Okay.
The basic idea is this. For some metabolites, if their potential dietary exposure falls below a very, very low threshold, we're talking micrograms per person per day, and they are not genotoxic, meaning they don't damage DNA, then they are deemed of no toxicological concern without needing a full separate toxicological workup. It's a scientifically validated way to prioritize detailed, costly assessments where they're most needed, not for every single tiny breakdown product that might exist at negligible levels.
Right, a pragmatic approach. Can you give us a couple of examples of where the TTC has been applied successfully?
Certainly. Remember, phlogocyproproline, some of its metabolites, even those with complex names like BCS-DE61185 and BCS-CC26101, were considered of no toxicological concern based on this TTC approach because their estimated exposure was so low.
Okay.
Similarly, certain phenpropitin metabolites, SYN515215, SYN515216, and SYN522621 were also assessed using TTC. And some prochloraz metabolites like BPS-4477 and BTS-5409-06 fell under the Kramer Class 3 TTC approach. It's a sound way to manage the sheer number of potential compounds.
Okay, moving on to real-world applications. This is where we see how all these scientific evaluations translate into the actual food you might buy and eat. We're talking about maximum residue levels, or MRLs, and how they apply to specific crops.
Exactly. Let's take azoxystrobin on melons. Based on studies looking at post-harvest use, an MRL of 5 milligrams per kilogram was estimated for melons.
Right.
And what's interesting is that this MRL was then scientifically extrapolated to watermelon as well. This shows how they can sometimes apply findings across similar commodities, which helps streamline the process.
Okay, that makes sense. Now, this next example sounds like it introduces a crucial, very targeted safety measure, particularly for women of childbearing age, fenpropidine on bananas.
Yes, this is a really important illustration. Supervised trials showed residues up to 6.0 milligrams per kilogram in whole bananas after foliar applications, even with a 0-day pre-harvest interval.
So right after spring.
Right. This led the meeting to recommend a new maximum residue level of 6 milligrams per kilogram for bananas. But here's the crucial detail. A separate specific acute reference dose, the ARFD, of 0.03 milligrams per kilogram of body weight was established specifically for women of childbearing age.
Why the specific group?
Because toxicological data indicated potential developmental effects, making that population group potentially more sensitive. It shows how granular these assessments are, recognizing that different populations can have different sensitivities or critical health endpoints.
That's incredibly detailed. Now, fipronil on rice saw a really dramatic shift, didn't it?
A major reevaluation, yes, absolutely. The maximum residue level for husk rice actually plummeted. It went from 0.4 milligrams per kilogram down to just 0.001 milligrams per kilogram. The asterisk indicating the limit of analytical quantitation.
Wow, that's a huge drop.
It is. And for polished rice, it similarly decreased from 0.15 milligrams per kilogram down to 0.001 milligrams per kilogram. These are substantial reductions, signaling a significant reassessment based on new data or evaluation methods, and this could potentially impact global trade and farming practices for rice, a major staple crop.
And pidoflumethofen on leafy greens presented a challenge you mentioned in setting group MRLs. Can you explain why it gets so granular for specific populations there?
Right, so they were considering a group MRL for leafy vegetables. However, the highest residue found in the trials for leaf lettuce was 6.5 milligrams per kilogram.
Okay.
When they calculated the potential short-term dietary intake based on that highest residue, specifically for children aged 1 to 6 in China, using Chinese consumption data, it exceeded 130% of the acute reference dose for pidoflumethofen.
Over the limit.
Way over the short-term safety limit for that specific group. And because of that exceedance, they couldn't recommend a group MRL that would cover leaf lettuce. It shows just how granular these assessments are, drilling down to specific vulnerable populations and their consumption patterns to ensure safety. One high residue on one crop type can prevent a broader grouping.
And when it comes to post-harvest treatment, like prochlorals on fruit, how does that residue change over time, say, during storage?
Yeah, they looked at that too. Data showed a decline in prochlorase residues on apples with a calculated half-life of about 58 to 69 days during storage. Similar declines were seen in pears.
So it breaks down over time.
It does, but interestingly, they also noted that the concentrations in the pulp of the fruit slowly built up over time, even as the overall surface residue decreased. It highlights that residues aren't static. They can move and change within the fruit itself post-harvest.
Now, here's a truly surprising fact that impacts, your actual home kitchen. What did they find out about phosmet and processing, like cooking?
This is really interesting for consumers, yeah. Phosmet was found to be only moderately stable under standard pasteurization conditions. But get this, it was found to be not stable at all under conditions simulating common home cooking methods like boiling, baking, brewing, or sterilization.
So cooking breaks it down.
Significantly, it seems. This is a crucial consideration. The processing and cooking you do at home can actually affect pesticide residues in your food, potentially reducing them quite a bit, at least for some compounds like phosmet.
That's actually quite reassuring. Finally, the meeting explicitly withdrew some previous MRL recommendations for cucumber regarding fulpet and for peaches and grapes regarding phosmet. Why would they pull back existing recommendations?
This happens periodically. It was due to either insufficient data matching the currently approved good agricultural practices, the GAs for those crops, or because the GAPs themselves had been updated and the old data no longer supported the MRL.
So things change.
Exactly. It underscores the dynamic nature of these regulations. They aren't set in stone. They are constantly being reevaluated and adjusted as new data emerges or as farming practices evolve.
Okay, to bring it all together, let's talk about harmonizing the global plate and the broader implications of all this research. What steps are they taking to ensure consistency across the world? Because different countries have different rules, right?
They do, and harmonization is a big goal. The report highlights the finalization of the classification of food and feed, which is Codex Document CXA 4/1989. This includes revised, internationally agreed-upon definitions for key terms like meat, muscle, and fat.
How does that help?
Well, having clear, consistent definitions is a really significant step because it facilitates the harmonization of MRLs globally. It aims for consistency in how we classify foods and measure residues across different countries, which should make international trade smoother and global safety standards clearer.
The meeting also recommended that all data submissions for MRLs be coordinated through a specific working group. Why is that important? Is it just bureaucracy?
Not just bureaucracy, no, it's really about improving efficiency. We're talking about an immense amount of complex data coming in from countless sources worldwide. By recommending coordination through the CCPR, the Codex Committee on Pesticide Residues Working Group, on priorities, they can better manage the flow of information, prioritize evaluations effectively, and ensure timely and consistent reviews. It's about making a complex system run more smoothly.
And the work is never really done, is it? It sounds like a continuous process.
Oh, absolutely. It's a continuous cycle. The report specifically mentions a multi-phase reevaluation strategy currently underway for a group of fungicides called dithiocarbamates. And they're also working on refining the guidance for interpreting toxicokinetic data. That's how the body absorbs, distributes, metabolizes, and excretes chemicals, specifically for the next JMPR meeting in 2025. It's a never-ending process of assessment, refinement, and improvement, always adapting to new science and new agricultural realities.
So we've really peeled back the layers today on how global science rigorously assesses the safety of pesticides in our food. It goes way beyond just spraying chemicals. It encompasses meticulous lab studies, understanding breakdown products, and even tracking real-world consumption patterns. It's this vast, intricate network of evaluation that touches pretty much everything we eat.
It truly is. And it's a testament, I think, to the incredible depth of analysis required, the constant need for reevaluation, and that intricate balancing act between scientific rigor, protecting public health, and acknowledging agricultural realities. Every single finding, every MRL adjustment, every ADI that's set or not set, it's a piece of this huge dynamic puzzle, all designed ultimately for our safety. It's like this quiet, ongoing revolution happening in food science behind the scenes.
So next time you look at your plate, maybe pause for a second. Consider not just the delicious food, but also this invisible network of global science working tirelessly behind the scenes to understand its components and ensure its safety. And it leaves you wondering, doesn't it? What new insights might the next JMPR report reveal about the foods we eat every day, and how might that change our understanding of what makes a truly safe, healthy meal?